Thank you. So I'd like to begin by thanking Aaron and Ronald for the invitation to speak here today and for sort of technical assistance this morning with this seminar. It's always a great opportunity to interact with my OTS colleagues, so I look forward to kind of interacting with you after this seminar. So I think for many years now, my lab has been interested in developing TNA molecules that we could evolve in the lab. And when we began this project, we wanted to create a class of molecules that didn't really exist, using an enzyme that also didn't exist, with chemistry that still needed to pretty much be worked out. So as you can imagine, this project has really been generational in my lab, with kind of each group of students working out kind of key aspects of the problem. So today, what I wanted to do is kind of tell you about our most recent efforts to create base-modified TNA aptamers, which we call threomers. Okay, these molecules are kind of conceptualized here on the right side of this slide, where we have the crystal structure of a base-modified DNA aptamer bound to a protein. And essentially, we would like to create very similar types of molecules, however, with the key exception that they would have a completely unnatural nucleic acid backbone. And in this way, we really feel that these molecules would have kind of all the best properties. They would have the solubility and the conformational flexibility of nucleic acids, the chemical diversity of amino acids, and the biological stability of an artificial synthetic genetic polymer. And really the goal of our work is to advance threomers to the point that they could compete with monoclonal antibodies as therapeutics. And of course, this is a really tall order because antibodies are arguably one of the most successful classes of drugs ever created. So they make up about half the entries on the list of blockbuster drugs every year. But despite this remarkable success, they do have limitations. Okay, the clinical benefit of antibodies is sometimes limited to a subset of patients due to variable efficacy or unexpected, acute, and sometimes life-threatening toxicities that are not always easy to predict. Even more challenging is the fact that monoclonal antibodies are cost prohibitive for the vast majority of people on the planet. So for example, if you look at Keytruda, it has a wholesale cost of more than $11,000 per dose. And of course, a patient on this treatment would be receiving a dose every, say, 3 to 4 weeks. So as a consequence, there's been a lot of interest in developing alternative strategies. And one approach that has been widely discussed is aptamers. Now, aptamers are much smaller molecules than antibodies. And you can see this on this slide, where the aptamer shown here is in orange, and the target is still kind of the same target you saw on the previous slide, kind of shown here in red. But if you zoom in on the aptamer, you see that it has many of the same properties of an antibody, okay? It's a single chain polymer that folds into a shape that can bind a target. And the footprint of an aptamer is generally very similar to the footprint of an antibody in terms of the surface area that it's binding. And when these molecules are properly designed, properly evolved, properly created in the lab, they can actually achieve very high levels of affinity and specificity for their targets. And relative to antibodies, aptamers have a number of important advantages. They're generally considered to be cheaper to discover and produce because they don't require animals for discovery and they don't require cells for production. They're also generally considered to be easier to scale and modify because they're amenable to chemical synthesis. Once you've identified your aptamer sequence, you can chemically synthesize it on very large scales. Now, even with these advantages, there are also a number of important challenges for aptamers that have existed for a long time. And these challenges are kind of outlined here. They're first and foremost the biostability. So biostability is a problem. So folks are starting to look for more and more kind of exotic backbone sequences. And once you do that, then replication becomes a problem because natural polymerases don't necessarily recognize those backbones. And then you have effectiveness, which is really the ability of an aptamer to bind a protein with kind of the same level of affinity and specificity of an antibody. Yes, aptamers can do that, but they can't necessarily do it every time. And so there's a lot of room for improvement there. And then the last one is throughput. Just the process by which you create aptamers is inherently very slow. So really what I thought I would do today is kind of walk you through these 4 areas and kind of tell you about some of the things that my lab has done to really advance the development of aptamers in these areas. So really the first one is stability. So the vast majority of aptamers that have ever been created are either DNA or RNA molecules. And of course, as soon as these molecules are introduced into a biological system, they can degrade in a matter of seconds to minutes by nucleases that are present inside an animal. So as a consequence, you really need a biological polymer that's much more stable. Okay, so there's been a lot of different work on different types of nucleic acid chemistry that has kind of evolved through a series of generations, but probably some of the most exotic ones are these molecules called xenonucleic acids or XNAs. And one of those examples is shown here. It's called threose nucleic acid or TNA. Okay, TNA was originally developed by Albert Eschenmoser during a period of time when he was exploring alternative polymers to RNA and really focusing on polymers that shared the same aldose sugar. So RNA is based on a ribose sugar, which is an aldose sugar, and there's a lot of other of those, one of which is threose. So this is a threose nucleic acid derivative of RNA, if you will. Now, these molecules are interesting. They have the same 4 genetic letters that are found in DNA, so A, T, G, and C, but they have this very unusual kind of phosphodiester backbone, okay? So the phosphodiester backbone is linked between the 2' hydroxyl group and the 3' hydroxyl group. And across the threose sugar, it's in this kind of unusual trans diaxial interaction. So we think TNA is a really interesting molecule for a number of reasons. One of the most important reasons is what I've alluded to already, which is its biological stability. So this is data that goes back quite a few years now, but it's an example where we chemically synthesized a series of different TNA oligonucleotides, and we evaluated their biological stability in different environments. So this is one example of that, where we incubated these TNA molecules in the presence of human liver microsomes. Okay, this is a very rich concentration of endonucleases and exonucleases. And what we saw is that TNA showed no signs of degradation during the period of time that we looked at. So this is plotted here up to 180 minutes, but in fact, the experiment was done up to 7 days. And even after 7 days, we saw no degradation. And this experiment's been reproduced in multiple different labs and even in companies. And it's really a striking result when you compare it to other sort of more common DNA and RNA analogs. So in this case, it's FANA and 2' fluoroRNA. So FANA is a DNA analog, 2' fluoroRNA is an RNA analog. These molecules are structurally very similar. They're just epimers of one another. And under these conditions, what you see is that these oligonucleotides with the same sequence as TNA degrade with a half-life of about 45 minutes. So there is quite a striking difference in biological stability of TNA versus some of these other molecules. Now, the other thing we really like about TNA is that it's capable of base pairing not only with itself, but also with DNA and RNA. And so TNA can self-assemble into an anti-parallel Watson-Crick duplex. And so shown here on the left is the NMR structure for a self-complementary Aptamer that was originally solved by Albert Eschenmoser with Bernard Juan. And then on the right, what we're looking at is an X-ray crystal structure of TNA cross-pairing with DNA. And in this case, the DNA is the black strand. This structure was obtained in complex with an engineered polymerase from one of my labs. And of course, you can also study the hybridization properties of these molecules using more traditional melting experiments. So this is an example of a particular duplex that was constructed with TNA and various TNA and DNA combinations. And what you see on the left is TNA self-pairing. In the middle is TNA cross-pairing with DNA. And on the right is TNA cross-pairing with RNA. And in each of these cases, you get these very nice sigmoidal plots where the inflection point represents the TM or the stability of the duplex. And if you look at it closely, what you see is that TNA base pairs more effectively with RNA than DNA. And this is something that's been well known in the literature. It's been demonstrated by lots of different groups. And actually, it was sort of looked into very closely by Martin Egley, using a number of very elegant structural approaches. And what Martin was able to find is that DNA is just less able to accommodate the constrained C4 prime exo sugar pucker of TNA relative to RNA. So RNA is much better able to accommodate that constrained sugar pucker that's observed for TNA. Okay, so that's how you could potentially solve the stability problem. And there's other XNAs that folks are working on. But regardless, once you've solved that problem, now you've got a bigger problem, which is how do you replicate those molecules? Okay, so ideally, if you want to evolve aptamers, you're going to need to be able to replicate them with enzymes. You're going to need to be able to take DNA libraries, copy them into XNA, and then at some point, copy those surviving members back into DNA. And so you could think about this using biological terms like transcription and reverse transcription. You could also think about this in terms of synthetic biology, in terms of information coding and decoding. But regardless of how you think about it, ultimately, it's going to require engineering polymerases because these molecules are not recognized typically by natural DNA polymerases. Now, there have been a number of different approaches to evolving polymerases in the literature. And the approach that we've developed in my lab is to do this in uniform water-oil emulsions that are created using microfluidic chips or microfluidic devices. And so kind of a high-level schematic is shown here of a little bit of the workflow. So, essentially, we engineer our polymerases at the DNA level. We transform those plasmids into E. coli. We grow and express the E. coli so that each E. coli is essentially expressing a different polymerase variant. We then encapsulate the E. coli inside a water-oil droplet. We lyse the E. coli, and that releases the polymerase as well as the plasmid into the solution. So you have this genotype-phenotype connection, okay? Now, if the polymerase is active, we have it set up in such a way that the droplet becomes fluorescent and we can sort that. So let's see what that looks like. So a full cycle of polymerase evolution is shown here, and we've dubbed this process droplet-based optical polymerase sorting, which we abbreviate DROPS. It's kind of a cutesy name since we're working with microfluidic droplets, okay? So this is a two-chip microfluidic process. So in the first step, the E. coli that are expressing your library of polymerases are encapsulated inside the droplets, but they're also encapsulated with all the other reagents that it would require to do a primer extension assay. So TNA triphosphates, the appropriate buffer, as well as an optical sensor. And so the optical sensor we use is a self-priming DNA template that ends in a 3' fluorophore, so it's a Psi3. And then we've got a short DNA probe that's annealed to that region with a black hole quencher. So as long as that probe is annealed, the fluorescent dye is quenched and the droplets remain dark. So at that point, once we've collected these droplets, we then heat them up to very high temperatures, so 90 degrees. This lyses the E. coli, but the droplets are stable under those conditions. We reduce the temperature to a more moderate temperature. In this case, it's typically around 55 degrees. That's because we work with thermophilic DNA polymerases. And then we challenge the polymerase to extend the primer to full length. And if that happens, then the quencher can no longer re-anneal and the droplet becomes fluorescent. If it does not happen, the droplet remains dim. Now we need to partition the active droplets away from the inactive droplets. And we do that on a second device called a FADS device. So this is a fluorescence activated droplet sorting device. Conceptually, it's very similar to a FACS system for cell sorting. So the droplets come down from the left, they pass through a laser, and you have laser-induced fluorescence that we can detect on a microsecond time scale. Then not shown here, but at the bottom is an electrode that can fire a series of pulses, which then change the dielectric, change the mobility of the droplet, essentially. It hits the divider and kind of rolls in. And so we can sort droplets that way. We recover them and kind of take them through another round of this evolutionary process. So this is kind of what it looks like. I'm going to pause the movie for a second. But the photograph on the left, what we're looking at is one of our PDMS microfluidic devices. You have two lines kind of coming in at the far end. So one line is delivering all the aqueous material, so your E. coli, your buffer, your sensor. The other line is delivering your oil phase. And then the line that's closest to us is the collection line where we're collecting the droplets. And so this chip is mounted on an inverted microscope that's connected to a high-speed camera where we can film these droplets being formed. And so this is an image taken from that camera. What you're looking at is a flow focusing device where the aqueous, again, is coming in from the left. The oil layers are coming from the top and the bottom. And it's a very stable device that can form these uniform droplets at a very high rate. So we're making droplets at a rate of about 35,000 droplets per second, or more than 100 million droplets per hour. And these droplets usually have an occupancy of about 10%, which means only 10% of them have an E. coli in them. And that's done to ensure that you maintain, on average, 1 E. coli per droplet. And they have a size of about 18 microns. Okay, so I'll pause that movie again. So once we've collected these droplets, we've lysed the E. coli, we've done our incubation, now we need to sort the droplets. And this is kind of a cartoon representation. Again, the droplets are flowing from the left. We have another stream of oil that's pushing them up so that if they're inactive, they just go to waste. And most of the droplets will go to waste. Occasionally, you will have a fluorescent droplet. It's going to pass through our sensor. It's going to trigger our electrode. It's going to change its mobility. And it's going to go into the collection bin. And that's what you're seeing down below. Now, this is a mock sorting experiment. It's not a real one. So the dark droplets have a dye in them, so we can tell that they're empty. And the clear droplets have a fluorescent dye in them, so we can tell that that they're a real fluorescent droplet. And that's how we can visually detect that. And again, they're coming from the left. Oops, sorry about that. And so when you see this, I'll pause it for a second. You see this droplet here. It's actively getting tugged on, which means it's triggering the electrode, and it will then roll into the collection bin. And it just happens kind of over and over again. And so this is a very stable system where we can just keep going, and we sort droplets at a rate, typically a rate of about 1,000 droplets per second. So, this process works. We've been doing it for many years now. I think other labs have adopted some of this methodology as well. And we've been able to evolve TNA polymerases. Now, what I'm showing you here is one of our probably first or second generation TNA polymerases. And what it is actually the crystal structure. It's going to be a movie of 5 crystal structures that'll walk you through each of the steps by which an engineered polymerase can synthesize TNA. Let's see here. So this is the apo structure. Oh, I should comment for a second here. Yeah, it's an apo structure. This is the thumb here on the left, the palms, the fingers in yellow, the N-terminal domain in purple, and in pink is the exonuclease domain. Okay, so as it comes around, it's going to bind to a DNA primer template. This is the kind of the binary complex. It's now going to bind to a TNA triphosphate. This is the open complex. We're going to close the fingers. You have catalysis where you add a TNA nucleotide, and you now translocate to the next position on the template. So it's really, I think, exciting to kind of see this with atomic level precision. This was a lot of work. It took more than 2 years to do this work, and it's over in like 60 seconds, but it's still fun to see. Okay, so over the time, we've kind of looked at different ways to evolve polymerases. And conceptually, what we've done is really shown here in this sequence fitness diagram. And this is just a cartoon representation. But largely, most of the work we've done is kind of representative of this orange dot here on this lower peak. We've explored a lot of different ways to create diversity into natural polymerase libraries, doing things like deep mutational scanning and other techniques. And by and large, those techniques introduce relatively small numbers of mutations into the polymerase. And, to a large effect, they just loosened the specificity of the active site of the enzyme. So you could get some level of TNA synthesis, but it was never really robust, okay? And so what we wanted to know is, well, how can we create much better TNA polymerases? And, could it be generalizable that you could do it for other systems as well? Ideally, we'd want it to be, maybe somewhere like this, up on this green dot, way up high on a peak that would have, much more TNA synthesis activity and perhaps even be much more of a specialized TNA enzyme. The question was kind of, how do you get there? And so, we explored a lot of different ways, but probably the way that's been the most successful to us, in recent time is doing homologous recombination. So this is really a technique that, excuse me here. Sorry, I bumped my, okay, so this is a technique that's really been around since the 90s. It was developed by Pim Stemmer. It's sometimes referred to as DNA shuffling, and when it's applied to enzyme evolution, it's called molecular breeding. Okay, and so the way it works is you have homologous genes. In this case, it's 4. So these are all B family archaeal DNA polymerases. Okay, you then add whatever mutations you want to, maybe improve the activity for a particular phenotype. So in this case, we added the RSGA mutations, which were mutations from our previously best polymerase. And then you also have regions that are 100% identical across the various homologs. Okay, that's represented by these bars. And then you recombine it. So we're recombining it using a more modern approach than what was done in the 90s. We're using PCR. So what this means is we PCR amplified each of these individual segments. So we have 11 segments and 4 genes. So that's 44 PCR reactions. So you essentially deconstruct the enzymes, you mix them all together, and you do one large PCR reaction, and then you reassemble it. And when you reassemble it, you've now shuffled all of the genes. But because it's done by PCR, you have some really interesting results. So you get all of the expected recombination products. So in this case, if it's 4 to the 10th, that's over 4 million variants. But you also get some novel recombination events because of template switching, which occurs during PCR. And you also get a few point mutations or single nucleotide polymorphisms. And that's just a result of mistakes that occur along the way. So the library diversity is actually much, much larger than what you might expect on the basis of purely recombination. Okay, so with this library, we then did kind of a pretty significant TNA evolution experiment using our droplet evolution. So we took this library, took it through 5 rounds of selection for TNA synthesis activity. After 5 rounds, we screened 500 clones, identified one that was a little bit better than the rest. It's called 5-270. These are really boring names. And what it means is it's the 5th round of selection. It just happened to be the 270th clone that we looked at. And at that point, we declared that a winner. And we then intentionally mutated it by error-prone PCR, took it through 2 more rounds, and the mutations were directed towards the thumb region, I should say. Took it through 2 more rounds, screened 100 colonies, identified one that we declared a winner, took it through 1 more round of directed evolution, screened 100, declared a winner. And then we mutated finally the palm and the finger domain, screened 100 colonies, and declared a winner. And at that point, we stopped to start characterizing because this particular polymerase was quite active at the time. So if we look at like all of the mutations that arose through this process, it's very significant. It's much more than what we were seeing by scanning mutagenesis and recombining mutations and things. And so we actually have 51 mutations relative to the wild type polymerase, and they're kind of color coded, and they're just shown here on the backbone structure of the crystal structure of COD wild type DNA polymerase. And so these 2 mutations in gray are exonuclease silencing mutations. We've got the 4 RSGA mutations shown in blue, so one here, and on the fingers. All of the gold mutations are the ones that arose through homologous recombination and were present in that clone that we observed after the 5th round of selection. And these mutations shown in red are 10 mutations that we observed through these 5 cycles of directed evolution. Now, how well is this polymerase? So when you evaluate the activity of polymerases, you typically do a primer extension experiment. And so you typically have a DNA primer that is labeled in one way. And in our case, it's labeled with an IR dye. It's annealed to a template, and you challenge the polymerase to extend the primer to the end. So in this case, we had our polymerase, our triphosphates, the appropriate temperature, and we extend it out. And if it works, you're extending it out with 40 sequential TNA nucleotides. So my mouse is a little sensitive. So when we do this, what we see is that our previous best polymerase, this polymerase known as COD-RSGA, and as well as some of our sort of evolutionary progenitors, if you will, have much weaker activity. Now, this is a primary extension assay. It was done under 2 minutes, and it's being visualized by denaturing polyacrylamide gel electrophoresis. So you have your primer. If the primer is extended, you've got more nucleotides on it and moves more slowly through the gel. So what you see is relatively modest addition. You get to round 8, you're starting to see signs of significant signs of full-length product. When you get to round 10, this 1092 variant, it is now very robust. So in 2 minutes, it can extend a primer all the way to the end with TNA. So if it can do it in 2 minutes, how fast is it really? So then you do a time course experiment. So this is a time course experiment measured in seconds. And what we see is 1092 can extend the primer with TNA in 45 seconds. So roughly speaking, you're looking at about 1 nucleotide per second, which places it within, say, 10 to 100-fold of the natural DNA polymerases from which this enzyme derived. Okay, and by comparison, our previous best is much, much slower, of course. We also see that 1092 is able to recognize TNA triphosphates much better. And so at lower concentrations of TNA triphosphates, we still see full-length product, where with our previous best polymerase, it's very marginal. We see that it's starting to give up its DNA as DNA recognition activity. So 1092 relative to, say, wild-type cod, it struggles to synthesize DNA at lower concentrations. And then finally, we see that the polymerase is also more accepting of base-modified modifications, base-modified nucleotides. So in this case, you've got C5-modified uracils that are being incorporated into TNA with phenylalanine and tryptophan side chains that were much more difficult to do with our older polymerase. And then finally, what we're looking at here is a thermal stability assay. So these are thermophilic polymerases. They're very stable. And so in this assay, we heat the polymerase at 90 degrees up to 6 hours. And at each time point, so each hourly interval, we take an aliquot, lower the temperature, and assay it for activity. And you see that the polymerase remains active even after 6 hours of incubation at 90 degrees. So it hasn't necessarily given up its extreme thermophilic stability, even though we've incorporated so many mutations into the backbone. And that could also be on how we do the selection as well. So, what does this structure look like? So we've solved the X-ray crystal structure of the closed ternary complex, and it's kind of shown here as an overlay with the wild-type cod DNA polymerase structure. So the 1092 is in purple, and the wild-type cod DNA polymerase structure is in gray. And what we see is some pretty major structural changes, especially in the thumb and in the palm regions, and they're kind of shown in these boxes B and C. And so if you look at it, you can see helices like this one right here, alpha helix 20, which moves more than 7 angstroms in the new structure. And likewise in the palm, if we look at alpha helix 12, it moves 3 angstrom, and it's also extended out further because it now contains an insertion, which came from one of our homologs. This came from deep-fent DNA polymerase. So lots of major structural changes in the architecture. Now what happens when we look at the active site? Well, this is kind of remarkable. The active site changes very little. So what we think is happening is the architecture on the outside is really fine-tuning the geometry of the active site. So you see TNA sits inside the active site pocket with a very nice fit. And if we just look at it in panel C, what you see is that the incoming TNA triphosphate is very coplanar with the base pair below it, and that the distance from the 2' carbon to the alpha phosphate is relatively similar to what we see in wild-type cod. The distance is here, it's 3.8. Now you're going from obviously the 3' carbon, because it's DNA, to the alpha phosphate, and that's 3.7. And again, it's very coplanar. So it's mirroring what we see in nature, as opposed to the polymerase that I showed you much earlier in that movie, which is called COD-RI. And that was one of our first generation TNA polymerases. You see the triphosphate really struggles to sit in the active site, that the distance between the 2' carbon and the alpha phosphate is very long. And that explains why this polymerase is so much slower, orders of magnitude slower than 1092. Okay, so now we've gone to the trouble of creating a TNA polymerase. Now we can begin to think about doing selection. So, a lot of the selections we do in our lab, we use a DNA display approach. So it's essentially a self-priming library. So this is all DNA that you're looking at. And once you have your TNA polymerase, you can copy the unpaired region into TNA. This gives you a very long kind of hairpin. We can then denature that hairpin, add a very long DNA strand, extend the DNA out, and now you've got double-stranded DNA helix that displays a single-stranded TNA molecule. Once we fold that molecule, we can then challenge it to bind a target, and this allows us to then create this genotype-phenotype connection. So if this aptamer binds to a target, we can then recover it. We typically do that by a photocleavage. So we photocleave the DNA, we isolate the DNA, we can PCR amplify the DNA, and then take it through another round of selection or sequence it. Okay, so that brings us to kind of the third challenging area, which is really binding. So a lot of aptamers tend to bind proteins with faster off rates. So you're looking at kind of a cartoon representation here of the kinetic profile of on rates and off rates of affinity reagents. So, we typically do BLI, but this could also be SPR. So you can imagine that this is your on rate, and then once you remove your target, you have an off rate. Okay, so most aptamers bind proteins with faster off rates. By comparison, antibodies tend to bind with much slower off rates, which is also why they're really good reagents in the lab, and they're also really good therapeutics. They bind and largely stay bound. So how do we kind of convert a faster off-rate aptamer into a slower off-rate aptamer, more typical of an antibody? And so one way to think about this problem is to look and ask the question, well, how did nature solve this problem? So you could look at crystal structures of antibodies bound to antigens. And if you peel these apart and you look at them in what you call the open book view, these yellow residues here are the residues on the paratope surface of the antibody. So these are the residues that contact the antigen. And of course, the yellow residues on the right are the residues on the epitope surface of the antigen that contact the antibody. But we're just focusing on the antibody for a minute. And if you look at these, you see a lot of ring structures, okay? And this is not uncommon. In fact, there's been lots of structural studies done looking at these interfaces. And this is one example of a study that looked at 53 different crystal structures. And what they found is that aromatic residues are about 5 times more abundant than other amino acid residues at the paratope surface of an antibody. So that's a huge clue. It means you need to add these residues to your nucleic acid libraries. Okay, so if you look at sort of the chemical diversity of typical DNA libraries, what you see is they're essentially heterocycles. They're aromatic rings that can stack well in a polar environment. They also have lots of nitrogens and oxygens, which means they're good at hydrogen bonding, but there isn't a lot of hydrophobic character. Okay, so how do you add that? Well, there's kind of 2 different strategies. One strategy is you add it after you enzymatically synthesize the oligonucleotide, and that's shown on the left. And on the right, the other strategy is you add it before you do the enzymatic synthesis, so it gets incorporated. So if we just look at the strategy on the left, this is oftentimes referred to as click Celex. It was originally pioneered by Isaac Krauss and Günter Mehr, very independently from their labs for different reasons. But essentially, in both cases, you're incorporating an ethynyl U derivative in place of the normal thymidine base. Once you synthesize the oligonucleotide, you can do a click reaction to then add your side chain to it. Okay, and this has been done many times by other groups. I know Marcel Holenstein, for example, created very nice DNA aptamers using cubane, which is unlikely to be a good substrate for a DNA polymerase. Now, the other strategy has been done also by lots of different laboratories. I'm just highlighting one example here, but this is in some ways a more technically challenging approach because you have to chemically synthesize the base modified nucleoside triphosphates yourself or find a way to do that. And then you have to ensure that they're substrates for your polymerase, okay? But again, this has been done many times by lots of groups. I'm highlighting one example here that's incorporating a lot of aromatic character. And so this is the chemistry that eventually gave rise to SOMers, the affinity reagents at SomaLogic that are DNA-based affinity reagents. Okay, so looking at these chemically modified nucleoside triphosphates, it was originally a lot of sort of trial and error to figure out which polymerases work best. But what folks have found is that by and large, B family DNA polymerases tend to work better with base modified nucleotides than A family polymerases. It's not exclusive, but just generally speaking, they work better. And Andy Marks really, I think, elucidated why that is by solving the crystal structures of a representative A family polymerase and a representative B family polymerase. And if you look at these structures, what you see is that the cavity in the major groove region of the active site is just much larger for a B family polymerase. So it can accommodate these different side chains than, say, an A family polymerase. So I think that's really insightful and very elegant work. So kind of recognizing all these advances in the field, my lab decided to go ahead and sort of take on the challenge of synthesizing base-modified TNA triphosphates. Now, TNA triphosphates are already difficult to make. Now making base-modified ones is even more difficult. So this is really, I think, a lot of work where we synthesized, in this case, 10 different C5-modified uracil derivatives of TNA triphosphates. Each one of these was 18 synthetic steps. So you're looking at like 180 synthetic steps to produce these molecules. We've also subsequently gone on and looked at base-modified cytidine TNA analogs. Again, they're C5 modified, and we've made 2 of those analogs as well. The cytidine analogs tend to be much harder to synthesize. There's a number of sort of strategic problems that you have to overcome when doing those. Okay, so what you're looking at here is data from 3 different selections done at different times by different people against different targets. But the important thing is that they're done with different libraries. So in the top row, what you're looking at is a selection against HIV reverse transcriptase with a standard-based DNA library. And what you see is, again, if we're looking at the kinetics of this, you've got your on rate and your off rate. We're just going to focus on the off rate for a second. You see a relatively fast off rate. Now, when you modify your libraries with like a phenylalanine in this case, and you select it, which in this case was the receptor binding domain of the S1 protein of SARS-CoV-2, you see the off rates are much slower. And then you could do, another example, in this case is with tryptophan modified libraries against the full S1 protein. And again, you've, very slow off rates. Yes, I recognize that the on rates are different here, but there are some nuances about HIV reverse transcriptase and how it just binds nucleic acids very, very well. So there is fast off rates here. I don't think it's necessarily representative of all protein targets. So the other experiment we did was we synthesized a TNA aptamer that we selected. So this is a tryptophan modified TNA aptamer that binds the S1 protein of SARS-CoV-2, and we engineered it to have 2 modifications. So now it's doubly modified. So all the U's have tryptophans and all the C's have phenylalanines. And we just did it just to see what would happen. And we see that, again, you have this nice slow off rate, and now the KD, changes a little bit, changes about 2-fold, suggesting that, doubly modified aptamers could be even more valuable. Okay. So I think, that's a lot about how you can improve the off rates of aptamers in ways we've thought about it, which are similar to ways other folks have thought about it as well. It just gets more challenging when you go into exonase. Now the last part of this talk is really about throughput. So historically, selecting aptamers has been a long and arduous task. That's because, generally standard-based aptamers are very rare in random sequence libraries, which means you have to do lots of rounds of selection. And that's conceptualized here. So essentially what you have is a cone that represents all the members of the library. So in this case, there's about a billion members, or a trillion members. So it's 10 to the 12. You have a fitness, so things that are members inside the library that are more active or better able to bind your target might be shown in pink at the top. And this green plane is really the threshold that you have to cross in order to be able to detect a binding event using an analytical technique. So they're very rare, which means you have to do lots of rounds of selection to enrich the population in binders. And again, that's a very slow process, and it's even slower for exonase. So the hypothesis we had is that, maybe we could accelerate this if we didn't use standard-based libraries, but instead used base-modified libraries. Maybe the base modifications, which enhance the function, enhance the chemistry, they enhance the binding, maybe they would enrich the population of molecules enough that you could actually find them from one round of selection. And that's something that we're really interested in. It's a little bit of a different approach to aptamers, but if it worked, it would accelerate the process. But this created a new question, which is then how can you evaluate discovery strategies? Because if you can do one round, you can do lots of different chemistries, you can do lots of different library designs, but you still have this bottleneck afterwards of characterization. And characterization of XNA aptamers is arduous. You have to enzymatically synthesize each one and purify each one, which is a very large scale transcription, which consumes a lot of triphosphate. It's really painstaking. So, can we change the way we do secondary screening? And so the idea that we had was to do this in a hydrogel particle format. So what you're looking at here is a microscope image of magnetic beads that are surrounded by a polyacrylamide gel coating. And these are all just made in bulk solution, but surprisingly, they're relatively uniform in size. And so this is really nice work that we did in collaboration with my colleague, Brian Pagel here at UCI. And so the way it works is, as you just add magnetic beads to an Eppendorf tube, you then synthesize the acrylamide shell. It's a very straightforward process. Once you have that, you synthesize them in such a way that your DNA primer is cross-linked in the acrylamide gel surrounding the bead. So this provides a more solution-like environment than a typical bead surface. And it's also very porous. So enzymes and templates and things can go in and out of the pores. So you can anneal your template, synthesize your TNA, remove the template, fold the aptamer. Now you've got folded aptamers that you've made that you never had to actually purify, and you can synthesize on a relatively low scale. Then once you've made those, you can do an activity assay, which is basically an ELISA assay, where if the protein binds the aptamer, or if the aptamer binds the protein, you can detect it using different, various fluorescent approaches. In our case, we have a biotin and a fluorinated streptavidin molecule that comes in to detect it. And once that happens, you can then assay the binding by flow cytometry, just flowing these particles through a normal flow cytometer and looking for shifts in fluorescence. So with that technology, we can now query libraries in a 96-well format. That's what we're currently doing. So you do a selection, you do one round, you just randomly pick sequences that you think have the right compositions. You synthesize 96 templates, and then you assay them in 96-well format. And if you get a binding event, you can read it out by flow cytometry by looking for shifts. And so this has really accelerated the characterization of aptamers. And it's now enabled us to do selections in parallel. So essentially, this is 6 different in vitro selection experiments that were performed against just a generic target that we were interested in. One was a standard-based chemistry, one had the leucine side chain, a cyclopropyl, a dioxyl, a phenylalanine, and a tryptophan side chain. So in this particular experiment, the tryptophan side chain worked particularly well. This is now published work. And so once you have a hit like this, you can ask other questions like, what if we look at different library lengths? So in the previous slide, we looked at 30mers. What if we had 40mer random regions or 25 or even 20? And in this case, we could still get, good hits from a shorter library. And that's important for us because eventually we want to synthesize these by solid phase synthesis. With this hydrogel assay, you can do a lot of other things. You can do specificity assays. So the aptamers that I was showing you bind the receptor binding domain of S1, which means they also bind the full S1 protein, but they really show no affinity to human serum albumin or streptavidin as a target. And so you can do kind of specificity tests. You can do lots of sorts of optimizations of your aptamers. So this is an end mapping deletion analysis where we can design templates that truncate the 3' end or the 2' end of TNA. Remember, TNA is a 3' to 2' polymer. So this is kind of what we would call the 5' end, if you will. So you can truncate one end, you can truncate the other end. You can do scanning mutagenesis by mutating, base modified residues to see if they lose activity. You can do base swapping. You can even do medicinal chemistry with this approach. So we really like this strategy a lot. It's kind of allowed us to put together what we think could be a nice pipeline for evolving XNA aptamers that could be done without evolution. So essentially sidestepping Darwinian evolution, getting to a point where you take libraries, you screen them against targets, you do modest sequencing, you screen your hits, you screen your hits. You then, once you have a hit, you can then optimize it using the same technology in these hydrogel particles. Once you have a really good hit, you can chemically synthesize it, even with the base modifications, and then you can validate it. And then once you have a validated hit, you can take it into functional assays. So this is just one example of a protein that came out of this pipeline. This happens to be an aptamer we selected to VEGF. And I think if you look at the BLI sensogram over here, what you see is an aptamer that's very representative of what you'd expect for a pretty good monoclonal antibody. So you've got an on rate, a very slow off rate. I'm sorry. And it binds with a KD of 150 picomolar. And it didn't require evolution. It didn't require directed evolution or in vitro selection. But it was brought through the pipeline. It was optimized the way I described. So I think with that, I'll just stop, and I'm happy to take any questions you might have. I know it was a lot of work, and we covered a lot of things. So if you have any questions that don't get answered, feel free to email me. Thanks.
Yes, thanks a lot for this excellent presentation. So maybe to start, so I think what we all want is we want to find a drug or a compound that works for a particular disease. So we want to deliver this drug, or these aptamers in this case, into human beings. So I think one of the challenges is the delivery. So within your developing pipeline, is there also a component that you can develop particular, I'm not sure how if you have to couple it to a peptide or whatever to deliver it within the cell, or to deliver them within the specific cell types, for example?
Right. So that's a great question. Delivery is always an issue for oligonucleotide therapeutics. And a lot of the way we've been thinking about it initially is to sidestep that problem by really just focusing on extracellular targets like cytokines, receptors, proteins that are in serum and things. And so, this would be delivered, if it was ever successful, of course, it would be delivered, ideally through a subcutaneous injection or through an IV, the way antibodies are largely delivered. But that being said, there are certainly approaches that folks have taken to deliver aptamers to the inside of a cell. This can be like receptor-mediated endocytosis. So you can target a receptor that then brings the aptamer inside the cell. That creates new complications, of course. You could think about, using it as an ADC, where you deliver a toxic reagent to a cell or a radioisotope. I think there's a lot of approaches there. I think if you were going to do intracellular targets, but you would want to build it into your selection strategy, that's kind of how I've been thinking about it. But certainly there's other ways to do it, whether you've got a peptide, another molecule, or a liposome or something like that. So thanks.
Yeah, I think a lot of people are working on these kind of strategies, so it might be nice to combine in the end.
For sure. Yeah, we're happy to work with folks on it.
Yeah, I think then the second question is also a similar question. Is always the off target. So how specific is this strategy? How to analyze whether there's any cross reactivity with these compounds?
Right, and I think that's something we need to do more work on. I think what we saw from our sort of hydrogel data is that it's recognizing its target very well, and it's not recognizing a small number, in this case 2, off target. So you can do those experiments in vitro, just in binding assays, by going after large numbers of targets. You could also do it on protein arrays that have maybe 1,000 proteins that you're kind of looking at, or you can start taking it into cells and looking for side effects as well. So I think that's kind of what we're thinking about that problem, but there is indeed more work that needs to be done. Our general hypothesis is that if it can bind its cognate target in the antibody range of kind of low to mid picomolar, it's largely going to be pretty specific, but that's just a hypothesis at the moment.
Any studies on the pharmacodynamics of TNA?
Limited. There's been limited. TNAs have gone into mice. They've not generated a significant or any noticeable immune response. We've done obviously the biostability studies against nucleases in serum and all sorts of things like that. But there's more work that needs to be done there as well.
Any to follow up next to the pharmacodynamics, any target engagement studies that have been done in animals already?
Not yet. Not yet.
That might also be interested to see.
Yeah.
So, if okay, I pass the word to Erin for the more chemical questions.
Great, yeah. So I have a question just kind of to start. One of the bottlenecks in selection of Aptamers, DNA enzymes, I would imagine it's similar with your technology, is how you translate from the sequencing data to actual candidates. So can you speak a little bit more on how you're narrowing down for your secondary screen from your sequencing data?
Yes. We put sequences up and we throw darts.
Okay.
No, it's not that easy. We're kind of agnostic on the sequences. It's because there's no rounds of selection, you can't look at enrichment like you would traditionally.
Yeah.
But keep in mind that we've done a lot of traditional selection experiments, and PCR bias can dramatically influence the outcomes of those selections. So even though something's enriched, you have to be careful about that. The way we pick sequences is we largely look at the composition of modifications, because modifications are what's really driving the activity here. And if you have too many modifications, these things will aggregate, they'll do weird things, or quite frankly, you will not be able to make them.
Okay.
Our polymerase is good, it's not that good.
Okay.
And so, you kind of have to be in these sort of sweeter spots, and we're empirically kind of determining that. But, it tends to be in like the 30 to 40% modified range tends to be the best. And we're starting with an unbiased library, so it's an equal distribution of A, C, T, and G. So it tends to get biased towards purines. The ones that work best tend to be biased a little bit towards purines and a little bit less on pyrimidines, which are the modified ones. And also, we obviously avoid long repeats of the same nucleotide.
Yeah, that makes sense.
Yeah.
Great. Okay, so just kind of to transition, another couple questions about the chemistry. So we have here, how stable is the TNA against nucleus compared to like LNA, MOE, and OME modifications?
In terms of nucleus stability,
Mm-hmm.
I think it's very comparable. MOE we've done less with. LNA we have done work with. MOEs are just more difficult to work with.
Yeah.
So, we've done work with them, largely chemical synthesis, synthesizing oligonucleotides for various projects. And we've done stability studies. Others have done them as well. I would say TNA, LNA, HNA, they're all kind of comparable in terms of their very, very strong nuclease resistance. MO, I just don't have enough knowledge on that one. It's always been so difficult to work with. We just haven't done much with it.
Well, that's fair.
Yeah, I would say Mo is probably in line with that, maybe somewhere. Yeah.
Okay, great. Some more structural questions. So how does the melting temperature of the TNA compare to that of kind of like RNA heteroduplex or a DNA heteroduplex?
Okay, so it's typically consistent on average with a DNA RNA heteroduplex. Okay. So it's very comparable. So that's ideal for the types of applications we're looking for. If you're looking for other applications, you might want things that have higher affinity to DNA or RNA, for example, like LNA.
Okay, great. So you did kind of talk a little bit about some other examples with mice and injections. This question is asking, can you comment on in vivo PK properties of a TNA modified aptamer versus these other traditional modifications, like your 2' fluoro and your O-methyl modified aptamers? And then the second part of the question is, do you know how quickly they're getting filtered out of a subcutaneous dose?
Yes. Okay. So again, we've had limited information there. We do not. Okay. So if you take a TNA molecule, you inject it, it's unmodified. You inject it, so there's no PEG or anything like that. You inject it into a mouse, it's cleared in about 24 hours. You don't see any major toxicities or anything like that. And the molecules are still intact. Okay. If you take other modifications like 2' modifications of RNA or whatever, they will degrade. And those metabolites oftentimes can create toxic profiles. So I think part of the hypothesis is that part of the safety of TNA is that it's not going to create those metabolites, and it's sufficiently sort of foreign that it's not being recognized by toll receptors and things. So, but again, there's a lot more work that needs to be done there, but it will clear unless it's modified and you increase the size. So, you'd have to do the traditional approach, like put a PEG on or make some sort of conjugate or something.
Okay. Okay, great. All right, getting into the kind of real specifics. So can you comment on the synthesizability of the building blocks and the TNA? For example, if you wanted to make like a 40-mer in a larger scale, potentially up to kilogram scale.
Yes, we can do it.
You can. Oh, great. That's so cool.
It's not
definitely.
easy.
And am I correct to say it's sequence dependent as well? Because you were saying some of them that are like highly modified will do weird things, get out, stuff like that.
There's some nucleotides that are harder to couple than others.
Okay, that makes sense.
Yeah, but it's a 2', 3' phosphodiester linkage, so that 3' hydroxyl, which would be equivalent to the 5' hydroxyl on DNA or RNA, is extremely sterically crowded.
Okay.
And it's a secondary alcohol versus a primary alcohol, so its reactivities change a lot in terms of removing the DMT and then actually doing the next coupling step. There's a lot of nuances there that you have to be aware of.
Fair. Okay, so moving on to the polymerase now. So do your engineered polymerases show promiscuous activity to other X-NTP substrates besides the T-NTP?
Very little.
Oh, cool.
At least on the ones that we've tested.
Okay, so now some of the questions are more kind of like general experimental design sort of thing. And so this person is asking, before you append a modified nucleobase or a heterocycle onto a sugar ring, what are some of the preliminary experiments that you would do? And do you assess the binding of the heterocycle to a DNA RNA strand?
No, we don't really do any of that. We're kind of an academic lab. We just make it and go. I think we've had, so many years of experience with these nucleotides. And I guess I would comment that the modifications are on the base, not the sugar. But the sugar is itself modified. So when you think about these base modified TNAs, the backbone is entirely modified. So the sugars are all unnatural because they're threose instead of ribose, deoxyribose. And now you've modified the bases as well. And so it's pretty unusual molecule. But yeah, you could think of it as like doubly modified, if you will. But largely the work that went into that and the thought process was first working out the methodology to chemically synthesize the monomers, and then second, working out how well they're recognized by whatever polymerase we had available at that point in time.
Great. So it is 12. Do you have a couple more minutes to answer 2 more questions, or do you need to hop off?
Sure.
Okay, great. So these are very general kind of like methodological questions. So this person is asking, aptamers contain 4 nucleotides, but proteins have 20 amino acids. How can aptamers make a structure that is as varied as proteins?
I'm sorry, how can they make a structure that is?
As varied as proteins. So
I think
structurally complex.
Right. I think the way we're doing it with base modifications, and it's a delicate dance because again, too many modifications, you can't make it or it does weird things. There are some structures of base modified DNA aptamers, and they're very interesting because they adopt different folded conformations than traditional aptamers. You get, there's one example where you have a three-stranded structure that's not helical. And so, you know, you can also have these sort of hydrophobic modifications making more protein-like structures where they create hydrophobic cores. So they have a structural role, but then they also have a functional role in terms of being complementary to the hydrophobic surface. So I really think it's a way to convert, conceptually, it's a way to convert nucleic acids into more protein-like structures. It's very interesting, and we're keen to solve the structures of some of our most recent aptamers.
That's really cool. I know that structural determination is also a challenge with traditional aptamers. And so that's really neat that you are going to take on this challenge to see what these structures are forming. So the last question here is a little bit of kind of into the future. So they're saying that you have these TNAs that are capable of binding to RNA. So do you think that it could potentially be used to silence genes, these sorts of molecules that are modified?
I only caught part of that question. So I think the gist of the question was, you have your TNAs also bind RNA. Could you think about using them for other applications besides aptamers?
Yeah, for example, gene silencing, yeah.
Yes, and they are. So I know at, you know, one of the most recent OTS meetings in Barcelona, there was a poster from Roche looking at TNAs for ASOs, antisense oligonucleotides. That there's been publications out of Alnylam looking at TNA as modifications that could be added to siRNAs. There's several groups in China that are looking at TNAs for different applications, gene silencing, also gene delivery by stabilizing RNAs that get delivered to cells by putting them on the ends. There's all sorts of things being done, and I think it's exciting to see that TNA is kind of percolating in these different areas.
Absolutely. Great. Well, John, I just want to thank you again so much for giving a really cool talk. We really appreciate you coming on and sharing kind of, every time I see you speak at something OTS related, it's a different story, which is really neat, that you're into kind of everything, it seems, in the functional nucleic acid world. And so just to wrap things up, I want to remind everyone that we have a couple more webinars lined up for the summer. So July 25th is Hassan and Jillian, who you'll recognize from the board of directors of the OTS, and they'll be speaking on nucleic acid therapeutics successes, milestones, and upcoming innovations. And then on August 22nd, we're featuring the Paper of the Year Award from the last OTS. And so we hope to see you there. Thank you so much for attending.